So this is a real pleasure that in the lockdown, it's been amazing being able to listen to so many wonderful seminars. And that's been a real privilege because I've learned a lot about sphingolipids, which in the past one would not have attended. So those are, I think, one of the benefits. So as Chris points out, I've really spent my large amount of my career understanding lipids. And I think I want to just say, I'm sure there are lots of students around, that actually lipids were not really very popular things to study. And it took a long time to convince people that lipids are important. And actually are becoming even, we are learning more about them because of the way they participate in so many both physiological and pathological functions. Okay, so next slide, if I can move. I'm having trouble moving my slide.
Just maybe click in the presentation. You might be, or try the arrow key, perhaps.
I've tried both. Okay. So phosphoinositides, they participate in throughout the cells. And if we start at the plasma membrane, we have a variety of phosphoinositides, particularly PIP2 and PIP3, that literally define the plasma membrane. Intracellular membrane compartments are defined by other different phosphoinositides. And I want to focus today on signaling and the role of phosphatidyl transfer proteins in this process here during phospholipase C signaling. I'd also like to touch upon the issue of not just controlling the levels of the phosphorylated forms of PI, but actually the regulation of phosphatidylinositol itself. And I want to recall then that the phosphatidylinositol synthesis, which is the parent lipid, which gives rise to all the phosphorylated forms, the PIP2s and the PI3Ps and so on, actually the lipid is synthesized at the endoplasmic reticulum. The third point I want to talk about is secretion from the Golgi, where the role of PI4P and PITPs are quite important. I think one of the most important themes that have emerged in the last 10, 15 years is the idea then that although lipids are synthesized at the ER makes contact with other membranes. So this is the concept of membrane contact sites. And most of the lipid transfer proteins tend to operate at these membrane contact sites. And indeed, the lipid transfer proteins themselves are what I call tether proteins that will connect the two membranes. So we have protein domains like the SMP domains, the org domains, the start domains, which are all lipid transfer domains. Of course, the one I'm talking about is the PITP domain. And I should point out that all these different domains are very, very specific in the lipid that they will transfer between these membrane compartments. So going back to the plasma membrane and PIP2. A PIP2 at the plasma membrane is actually the most amazing lipid because more and more functions of PIP2 are being discovered. So of course, the requirement of PIP2 for cytoskeletal reorganization, for ion channels, it's a requirement for exocytosis, endocytosis, recruitment by proteins using domains, particularly the pH domain. So in all of these contexts we're looking at is the lipid is actually required in its intact configuration. There are two pathways where it's actually required as a substrate. It's the PI3 kinase signaling pathway and the phospholipase C signaling pathway. And it's really the phospholipase C signaling pathway where the consumption of PIP2 and therefore its removal is very, very significant. And I just want to say a few words about phospholipase C signaling. Phospholipase C signaling was originally identified as a mechanism for producing second messengers. And of course, this is now in the textbooks, IP3 mobilizing calcium, diacylglycerol activating protein kinase C, and other effector proteins. What is important is that the amount of lipid that needs to be hydrolyzed to saturate these responses is actually quite small. And I call those activators weak activators of phospholipase C, where the drop in the PIP2 levels will be not that great. Therefore, to restore homeostasis will be easier. But we also have another set of activators, which are called strong activators, where the phospholipase C activation is strong, is maintained, and the depletion of PIP2 can be very, very high. And the reason you need this high depletion is because you're not trying to make IP3 and diacylglycerol in this case, but very often you're wanting the PIP2 change to regulate ion channels. And the most famous one of them is the potassium channels, the KCNQ channels, which are best analyzed in that the PIP2 keeps it in the open state. Activation by the muscarinic agonist, which is a strong agonist, you can deplete as much as 30% of the phosphoinositide pool, and you get channel closure. And of course, to reopen the channel, then you've got to restore the PIP2 levels again. So having given you an idea that we need to think of phospholipase not as just a simple reaction, but really the strength of the stimulus will determine how much lipid is hydrolyzed. So here is a version of showing the cycle of reactions that take place. So here is PIP2, and here is the phospholipase C. And of course, this is a regulated enzyme by receptors. It's also regulated by calcium. Remember that there are altogether 13 different phospholipase Cs in the human genome. Nonetheless, the important thing is that the plasma membrane contains the machinery to synthesize the PIP2 from PI. So the lipid kinases, PI4 kinase, and the PIP5 kinases reside at the plasma membrane. The product of phospholipase C is diacylglycerol, which is actually rapidly converted back into PA. The machinery to resynthesize PI resides in the ER, and for this you have the conversion of PA to this intermediate CDP-DAG. There are two enzymes that will do the job. We will come to that later. And a single enzyme, PI synthase, to convert back into PI, and a reaction that requires inositol. It is this two arrows, which is the issue of how do we transfer PI from the ER to the plasma membrane? And secondly, how do you transfer the PA to the ER for reuse of that PA generated through the cycle? And initial studies had identified PITPs, which were able to transfer PI. Let me explain how this was done. So we're talking about methods. We use permeabilized cells to reconstitute cellular function. This is very gentle permeabilization, where most of the cell has holes in the membrane, but are still completely functional as far as lipid metabolism, signaling, secretion is concerned. So it's a fantastically good model system. And effectively, what we've found that if you take cells and you stimulate them in the presence of the cytosol, phospholipase signaling is brilliant. When you deplete them of cytosol, you lose phospholipase C signaling. Despite the fact that we already knew that the G proteins, the receptor, the phospholipase was still present in the system. Using this permeabilized cell system, we could reconstitute function by just adding back brain cytosol. And of course, several years later, after lots of time in the cold room, we purified the protein that was able to restore function, and we identified the first PITP, PITP alpha. Now, what is PITP alpha? Effectively, it's a bucket, and in the bucket, it's loaded with a lipid. And in this bucket, you can load itself with two separate lipids, phosphatidylinositol and phosphatidylcholine. And if you look at this structure here, when the lipid is loaded in the protein, you cannot see the lipid at all. Lipid transfer occurs in the absence of ATP, and you don't really need anything. You just need two membranes, one PI pore, and it will move it in the opposite direction. Okay. So after the first PITP was discovered, where 5 other proteins or 4 other PITP proteins were discovered in the human genome. So today we know that there are 5 of these proteins. And I'm talking about these ones, the small ones, PITP alpha and beta, which are effectively a single domain protein, which are able to then transfer lipid. But then you have these longer forms. And here, I think nomenclature becomes a bit of an issue. So they are either known as RDGB proteins, and that is because the first of these multi-domain proteins, RDGB alpha, was actually found in Drosophila and was required for phototransduction. And in the absence of RDGB alpha, you got retinal degeneration. So we will come back to that later. The RDGB proteins are also known as near proteins, or the gene name is the PITP-NM1 proteins. Okay, now we know how the PITPs function in terms of how they are loaded with the lipid. And it's an important point to make here is that the inositol head group is really embedded right at the end of the structure, and the tails are sticking outwards. So there is no way that within the structure, the head group can be phosphorylated. So the lipid has to be dropped out onto the membrane before phosphorylation of PI can take place. That's point one I wanted to make. The second point I want to make is that the inositol ring makes contact with 4 amino acid residues. And these 4 amino acid residues are literally present on 2 of the beta sheets, so they're not in a single row, but they are really a hallmark of all PITP proteins. And if you do a bioinformatics analysis of, there's now several thousand PITP molecules present in different species, you find that these residues will be completely conserved. Not only are they just conserved, but mutation of any one of these residues will lose loss of PI binding or PI transfer. As we will see later on, that this can then be used to ask the question, how important is PI binding? So I want to basically explain that we've grouped these PITPs into 2 groups, class 1 and class 2. And this is based not only on their sequence analysis, but it's actually also based on the fact that it turns out that what I call class 1, this is the original PITP that people had worked on, transfers PI and PC. So these are PI-PC exchange proteins. In contrast, it turned out that the class 2 PITPs are binding PI and PA. So if you think about it, it's these class 2 PITPs which would be, if you like, in phospholipase C signaling, would be able to do the reciprocal transfer of PI versus PA. And so we really wanted to examine this, and we took advantage of looking at the fly system, the phototransduction system. The fly phototransduction system is very specialist in terms of phospholipase C signaling. And that kind of makes it a very useful system to study in vivo, if you like, in a live living fly, how these proteins actually function in maintaining phosphoinositide levels in that system. So in order to do that, I teamed up with Raghu Padijat, who is, at that time when we started this work, he was in Cambridge in the UK. And he basically was really studying the phototransduction pathway. And we were able to use the fly system to ask the question, using the mutants, which were defective in PI binding, whether this binding of PI actually was central to function. Now remember that in the fly system, the RGGB protein that we're looking at is only required for phototransduction and also for olfaction. So effectively, if you make flies without RDGB, the flies are completely viable, they're fertile, they can do their thing. So it makes it a simpler system to do in vivo analysis. So let's introduce you to the fly system. Incidentally, humans don't use phospholipase C transduction. We use cyclic GMP. But we do lots of phospholipase C signaling in the brain. Here is the fly eye. It's made of ommatidia. Each ommatidia here has got 7 photoreceptors. So they're called rhabdomeres. And each photoreceptor cell has got this region, which is the rhabdomere. And that is where the visual machinery is. The rhodopsin for light, the G protein, GQ, phospholipase C, and the enzymes, the lipid kinases are all localized in this membranous structure here. In the fly, it was well established long before people were talking about membrane contact sites, was that the ER, a specialized form of ER, known as the Subtomeric Cisternae, is essentially the ER, was localized absolutely underneath the rhabdomere region. And this was established by Suzuki et al., beautiful EM data showing that this is only about 20 nanometers apart. So classic membrane contact site. And of course, what I'm looking here, what the red is the phalloidin, because we know that the microvillus is very rich in actin. So you can stain it for phalloidin. And if you then stain for the RDGB proteins, you can see that they lie just underneath that region there, in the subrhabdomeric region. So the protein is localized at the rhabdomere region. And we know how it is localized because the RDGB protein has got a binding site for VAP, and VAP keeps it at the ER. So what are the phenotypes that you can see when you take away RDGB alpha? You get loss of the light response. So this is the light response that you can monitor. And in the knockout, or you get, this is actually a hypomorph, so you lose most of the response. You get retinal degeneration. And what you mean by that is that the actin cytoskeleton has disappeared, and that's because PIP2 levels have dropped. And the proteins, the ERM proteins, which maintain the cytoskeleton, has disappeared. So this is a very classic phenotype of in the absence of RDGB. And of course, you get loss of PIP2. So these are flies which are now expressing a pH domain fused to GFP just in the photoreceptors. And you can see the photoreceptors there. Incidentally, there are 7 photoreceptors in an ommatidium. Only 6 are responsive to the light that we use. That one responds only to the UV light, and hence you can still see it there. So when you shine a light, you can see the PIP2 disappear. And if you wait long enough, it will come back again. And if you look at the RDGB mutant, you can see that the PIP2 has pretty much already gone down in the resting fly, because these flies have not seen any light at all. And as you can see, there is very little recovery. And you can restore function by re-putting back the full-length RDGB protein. So that's the protein restored function again. So there are 3 phenotypes we can monitor: the light response, retinal degeneration, and PIP2 levels. Now, one of the observations that had been made quite earlier on is that you don't need to use the full-length protein, the RDGB full-length protein, to restore function. You could literally restore function just using the PITP domain. And that told you that actually the functional part was the PITP domain, and the rest of the molecule was required for tethering the protein between the plasma membrane and the ER. So we've used in this experiment now just the PITP domain. And we've then used all these various mutants that we could generate. And you can easily see this is the control, this is the knockdown. You can't restore using any of the mutants that I described earlier on. The mutants I identified here. So whether you're looking at restoration of the degeneration, or you're looking at restoration of the production of IP3, which is an experiment that you can do in the permeabilized cells. Effectively, from these results, the binding of PI and the transfer of PI was absolutely essential for restoration of function. That applied the same to restoration of PIP2 synthesis. And here is an experiment here where this is the mutant added there. And here is the rescue now with a full-length mutant, which is just a point mutant here, K61A, compared to the rescue with the wild-type protein there. So again, just a single point mutant, which can't bind or transfer, you lose phototransduction function. So effectively, what we could conclude from these studies is that the RDGB proteins, what I call the class 2 proteins, effectively could reciprocally move PI in this direction and PA back to the ER for synthesis to take place. Now, the question was, if we could take the PITP domain of class 1, which only transfers PI and PC, would that work? Or do you really need to have both PI and PA transfer embedded within the same protein? And interestingly enough, you do need a PITP that transfers both PI and PA, because if you use a class 1 PITP, it's basically ineffective for either retinal degeneration or the light response. So as far as the fly studies were concerned, you needed a PITP that was able to move both PI and PA together. The second question we wanted to ask was, it is obviously an assumption that in vivo, this lipid transfer protein is acting by transferring lipids. In other words, the protein is actually going through an open and closed conformation and allowing a lipid to be dropped out, a lipid to be added in. So to address that question, we were able to take advantage of another set of mutants, and it is the mutant which are to do with these two residues here, two tryptophans. And we realized that these two tryptophans, in studies that we'd done, that had identified that if you mutated those residues, the protein was unable to do any lipid transfer. And we took advantage of this by using the cysteine residue, which is only exposed when the protein is in the open conformation. And so if you treat cells with anything that will modify the cysteine residue, you can observe this open and closed conformation taking place within cells. And what happens is that when the protein is trapped in an open conformation by docking a large molecule like NEM there, then this lipid transfer protein is going to be stuck on the membranes. It requires these WW tryptophans for it to take place. And it can no longer pick up a lipid because it's the site is already occupied by something else. And therefore, you can actually capture the lipid transfer protein at the plasma membrane or at the membrane. So you can see that these two residues are really, really important for lipid transfer. So just gives you a model of how we envisage things to happen. The tryptophan residues make contact with the membrane. It allows the movement of this, what we call the exchange loop, outwards, and this allows this helix to swing out, opening up the cavity, exchange to take place, and for the lipid then to move away. So we wanted to test this again in the fly system, because in the fly system, again, these residues are present, and the residues present are mutated. Lipid binding is unaffected, but lipid transfer is diminished by 90%. And you can see that if you do rescue with this mutant, you can't, and neither can you get back the light response. So again, lipid transfer is a very important part of the function of this protein. So effectively, I think we identify the RDGB class of proteins as being very important in phospholipase C signaling, particularly working at membrane contact sites. Because although we know that we can use just the PITP domain for reconstitution, we also know that if you use the full length protein, it's basically provides a better signal. In other words, when you use a very bright light, you really then need to have this protein sitting at the membrane contact site. Okay, this is a quick summary of that in Drosophila, the RGB alpha is required for phototransduction. Actually, the same is true in C. elegans. The single RDGB protein is required for chemosensation, olfaction, and again, it's phospholipase C signaling. And again, you can just use the PITP domain on its own. And interestingly, it's only present in very, very specific neurons. Zebrafish, again, the single protein, actually in this case is lethal because it has an impact on lots of brain activity, and there is a lot of issues both in the brain, both in the cerebellum and so on. Interestingly, in mice, well, we have two RGB proteins. The mice are actually quite normal and fertile, but really nobody's really done real analysis of any brain function, except with the isoform, the second isoform, what I call PITPNM2. But its expression is actually present only in a small area, the retina and the hippocampus. And here we know very clearly that it's required for visual low light conditions when you have very dim light and you have these photoreceptive cells. These are ganglion cells. And you really, if you don't have the PITMM proteins, then you have defects in circadian photoentrenchment and your pupillary light response. So you won't change the shape of your eyes. So actually, nobody's really done much studies really with the PITPNM1 or the NER2 proteins in any model animal systems, in mice, for example. Okay, I want to just go back to the soluble PITPs, because that's where we started with phospholipase C signaling. And the question was still there, whether these were still involved in phospholipase C signaling. And the answer actually is yes. In this case, if you knock out PITP alpha or beta from mice, it's bad news. It's lethal. You can't knock out both of them. So what we've done here is knock them out specifically in platelets. And these are experiments I've done in collaboration with Charles Abrams in the US. And so this is a knockout of PITP alpha. And you can see very clearly when you look at platelet function, IP3 production is greatly reduced, and as is the calcium response. So platelets are different. They need these PITP alpha and beta function. And here we've knocked out, that's alpha, this is knockout of beta. And you can see that if you knock out beta as well, you get the same problem. Less IP3 is formed. Now, an important point I want to make here is that we've quantitated the amount of alpha and beta in platelets. Alpha reconstitutes about 80% of the PITP. Beta is literally only about 10 to 20%. And yet, even removing, so this is removing beta alone, you get a big reduction. Knowing that under those conditions, you've got plenty of PITP alpha present. And that's kind of surprising because it tells us that somehow it's not working in a linear fashion. Okay, so just a small amount of PITP beta present seems to be sufficient for this process to take place. And here really summarizes it in a way that if you knock, this is looking at the amount of lipid transfer activity in the cytosol. So this is the wild type. And if you knock out alpha, you get very little transfer activity left. If you knock out beta, the drop in transfer activity is really low that it's not really measurable. However, when you knocked out both of those, you lose everything. So I think there's some, so in addition, if you like, of just concentration-dependent lipid transfer, I think that the transfer proteins are doing some additional function to be able to account for this result here, that in the absence of just 20% of a PITP molecule, you get quite a large reduction. Okay. So going back, why does the fly photoreceptor use a protein with a tether, if you like, a protein that's associated with the ER to allow a function at the membrane contact site versus something like a platelet? And I think it's to do with size. A fly photoreceptor is really, really big. And so if you are going to use a small 35 kilodalton protein, it's going to be very inefficient. But as far as a platelet is concerned, which is only about 2 micrometers, then maybe you don't really need for it to function at the membrane contact site as much. And therefore, a small PITP can do the job. And maybe a platelet doesn't really care about restoring homeostasis in a big way, because this is a long-lived cell compared to a platelet. A platelet is basically turning over very rapidly as a kamikaze cell. So it would appear that context is very important in terms of understanding which of the PITPs function in phospholipase C signaling. I want to very quickly shift gears and touch on the problem of the synthesis of phosphatidylinositol itself. And the reason is that during very intense stimulation, you can reduce the phosphatidylinositol levels by as much as 30%. And the question is, what mechanisms come into play? What pathway is upregulated to be able to allow the cells to maintain their PI levels? And effectively, what we know that there are some receptors that do not get desensitized. So this is the case of the vasopressin receptor. You can treat cells with 24 hours. In this case, you get differentiation of a cardiomyoblast into a cardiomyocyte. You can see this hypertrophic response. And under those conditions, the cycle is really driving the hydrolysis, and it would appear that some of the components may not be reutilized, and therefore you would need to drive more PI resynthesis to take place. And I'm going to push that forward and explain that what's really going on is during phospholipase C activation, protein kinase C gets activated. Long-term stimulation of CFOS drives expression specifically of all the components that we checked. It drives the expression of CDS1. So it's the CDS1 that seems to be important for maintaining PI levels during intense signaling that takes place in cells. So I think we've had enough of phospholipase C, and I want to just move on to the last of the PITPs, and that is called PITP-NC1. Very little work had been done on this PITP. And the first thing to note is that there are two splice variants. One's got a very long tail, and one's got a short tail, which looks more similar to these PITPs. And we decided to use zebrafish to analyze the function of PITP-NC1. And what is interesting, that in mammalian cells, you've got splice variants. So this is the splice variant 1 on the top. That's the long form there. And this is the splice variant 2, the short form there. And firstly, I want to point out that when cells get differentiated, there's an increase in both the splice variants. Here is the neuronal cell, get an increase in both. In zebrafish, instead of having splice variants, you have two separate genes expressing these two proteins. And the long version, which is basically the homologue of the long form, is only expressed in the brain. So you can see expression in the brain and nowhere else. And that made it very easy to ask the question, what was the function of this particular splice variant? Now remember that PITP-NC1 is a lipid transfer protein for PI and PA. But what I haven't really said is that its ability to bind PA is extremely high. And we know that the PA that it likes to bind actually comes from phospholipase D. So this is studies that we've done in are permeabilized cells that when you stimulate phospholipase D, the PITP NC1 proteins will grab the PA so that actually, instead of binding PI, most of the protein will be bound to PA. Okay, so what happens in the zebrafish? Effectively, the zebrafish larvae are hyperactive. Okay, they don't sleep at night very much. So these are the ones there, the red line, compared to the wild type. During the day, they're hyperactive. During the night, they're hyperactive. So they have a sleep problem. Not only do they have a sleep problem, if you look at their brains, this is under control conditions, and this is the knockout. This is expression of CFOS. There is a lot of neuronal activity already taking place in the knockout mice. What we know is that we can reverse this if we add back an inhibitor of IGF signaling, IGF-BP2. So effectively, what's happening in these knockout mice is that we have increased IGF signaling, and therefore this increased CFOS and increased activity. All of this can be reversed by inhibiting the PI3 kinase pathway. Now, whilst we were doing this work, a paper was published by a group in the US where they had identified that PITP NC1 was promoting metastasis. And it did so by recruiting Rab and allowing malignant secretion. And effectively, their model system was that PITP NC1 would bind PI4P, according to the cartoon. And it will recruit Rab, which will somehow produce PI4P, recruit Golf, and bind to this myo-18-1A and allow secretion to take place. I think that this idea that PI4P will bind in the binding pocket is very unlikely based on the structure here. This is a model structure. I think this is definitely a PI binding protein. We know that it binds to 1433, again, at this end rather than at this top end there. And nonetheless, we were trying to understand how we could rationalize our observations in the zebrafish in terms of the context of the secretory phenotype. Because I think our suspicion was that PITP NC1 was allowing production of PI4P for vesicular traffic to take place to the plasma membrane. And so the general idea was that PITP NC1 was allowing the secretion of IGF-BP2 in neurons. And this was putting a brake on IGF-1 signaling. And when you took away the PITP NC1, you get increased IGFR signaling and the phenotype that we're looking at, the increased neuronal activity and the waking. And sure enough, it was clear that PITP NC1 certainly regulates the secretion of IGF-BP2. So I think the PITP NC1 very likely functions at the Golgi for maintaining PI4P levels, for allowing secretion of a particular set of cargo from these set of neurons. And so in summary, I think PITPs are very versatile molecules, and the different members participate in really quite a range of physiological functions. Not terribly surprising because phosphoinositides do so many things. But I think what they do is they provide PI to different compartments for phosphorylation. And this provides unidirectional flow of PI from the ER to other membrane compartments. Remember, there is no ATP requirement here. I think the role of RDGB alpha, at least in the fly system, is very clear that it's required for phospholipase C signaling. And I think our prediction would be that in mammalian systems, it's pretty much doing a similar thing as well. These can also function in PLC signaling. I haven't said that, but the PITP alpha and PITP beta are very likely, particularly PITP beta. It does localize at the Golgi. It probably has functions at the Golgi in maintaining PI4P levels in the different compartments. Levels of PI are regulated by the CDS1 enzyme, not CDS2. And I think probably a lot of the transfer proteins will participate in membrane traffic from the Golgi. And I want to end by the fact that the infection by viruses depend on PITP. And recent studies using even the SARS-CoV-2 virus requires PITP-NM1. So effectively, what is known is that the replication organelle that the viruses use need cholesterol, and they get the cholesterol using the oxysterol binding protein from the ER. And of course, this means that the PI4P has to be transferred back to the ER. And to maintain this gradient, you need the PITPs. So PITP beta is being identified for some of the viruses, and in some cases, PITP-NM1 is. So on that note, I would like to thank some of the people who've done the work, my funding agencies, the British Heart Foundation, the Wellcome Trust, BBSRC, the fish work done by Tim Ashlin, who's now working at Glaxo, in collaboration with Jason and Marcus. Nick, very much involved in the studies with the CDS enzymes. Catherine Garner, who's now a lecturer in the north of England, is very much was involved with the work on PITP-NC1, as was these other people. And of course, the fly work, and I spent a delightful period as a sabbatical in Bangalore with them. And it was amazing to be able to work with a live organism. And the work on platelets was in collaboration with Chase Abrams and Liang Zhao. Thank you very much.